Bonus episode 87.5 Protein Stringy. Hello and welcome to this bonus episode of the history of American food. This time I'll be dusting off my chemistry teacher coat and explaining about proteins and how their different properties contribute to how these molecules work in food. So put on your safety goggles, pull up to your place at the lab table, and no eating, or you might turn into a dinosaur. The first thing you need to know is that most people in the 18th century had no idea about the chemistry of protein, but everyone who was involved in harvesting, processing, and preparing foods had opinions about protein-containing stuff. This episode is mostly going to look at protein from a food lens and definitely not a nutrition one. That's an area I don't want to get into, and anyway, nutrition as a science doesn't even begin to be a hot topic until the end of the 19th century. If you don't know any of the chemistry either, well, here's a chance to learn some practical chemistry and maybe understand why some cuts of meat are chewier than others, why jellyfish are like that, how protein relates to umami, whether mushrooms even can be a good source of protein, and why people, for ages, have successfully lived mostly on bread alone. So the big questions: what is protein? Where is it found? What does our body do with it? The basics, not the details again, not my area of expertise. And what does protein contribute to the American pantry? Proteins are made up of little puzzle piece units called amino acids. There are at least 500 different amino acids out there, but human bodies only build things with about 20 of them. These same 20, with just a few additions and exchanges, are all the amino acids found in the vertebrates in the animal kingdom. The insects have a few more. The single-cell organisms have some more as well, but the plants, they have the most more. All sorts of amino acids they're using. Because we're talking Americans and we have mostly restricted ourselves to eating vertebrates and plants, I won't get particularly exotic. All amino acids out there have 3 important ends and a little hydrogen nubbin. There's a central carbon atom that's the center of every single amino acid. When amino acids connect together to make the chains that eventually become proteins, it helps to think of them as having 2 arms that connect into a chain, end 1 and end 2, and then end 3 is the functional group, what's known technically as the R group, and that's where all the action is. And then because it's a carbon, which needs 4 connections, there's a hydrogen capping off the 4th connection. It's just like a little nubbin. So end 1 has a nitrogen end with 2 hydrogens. This is the ammonia part that gives amino acids the amino part of their name. Ammonia, that stinky, eye-watering cleaning chemical, is a nitrogen with 3 hydrogens, and it's pretty stable, but also very ready to take part in energetic reactions. It's the basic end of the compound for protein chain bonding purposes, but I'll stop there because we get into ammonia way more when it's time to talk chemical fertilizer and World War I. The second end is the carboxylic acid end, or COOH. This is the acid part of amino acid. Carboxylic ends are found in what are commonly thought of as organic acids. That is, they have big, long carbon-hydrogen-oxygen chains connected to a COOH on the end. And they tend to have jobs in or are waste products of living organisms. Some familiar examples include formic acid, the chemical that gives ants their sting, acetic acid, which is vinegar, and propionic acid, which is part of that particular funk of BO. And most of the long-chain fatty acids that make up fats, and I'll get more into that with my fat molecule bonus episode, are nearly all carboxylic acids. There are so many more, but that gives you an idea that the COOH ND on molecules is important in holding the living world together. The third end, as I said, is the action end. It's where all the excitement comes in. This R group can conceivably be just about anything. Some of the exotic ones get beyond just carbon, hydrogen, and oxygen and regularly include nitrogen. There's one that includes sulfur and one that includes selenium. Amino acids are used in 2 ways in living systems. They can be linked together to form larger compounds, but individual amino acids are also used as chemical signals, either as they are or after further modifications in the body. The ones used for signals and other stuff are usually what are called non-proteinogenic acids. They don't end up getting used to form proteins. They're made by special alterations of the 20 standard biologically produced amino acids. Humans can make 11 of the 20 essential amino acids from atoms and molecules roaming around in our bodies. For 9 of them, we need inputs from the outside world, that is, food, to make our basic complement to keep the works running. So when you hear the phrase complete protein, what people are really talking about are foods that contain the 9 missing amino acids. Funny story. Some of the classic combinations of food humans have come up with out there in the world fill this gap. So milk proteins and wheat bread, so yogurt and naan, for example, make up the 9 missing amino acids. So do rice and legumes, so red beans and rice or rice and tofu. And in the world of corn, pepitas or squash seeds, along with small bits of fish or bird meat, can provide the missing amino acids. But much more fun and full circle is the fact that corn can bring its own solution with it. There is a fungus or mushroom known as huitlacoche that infects corn and has truffle-like meaty flavors. It's very fashionable right now. And it fills in the gap for the missing amino acids when eating corn. So corn, once again, is proving to be the most borderline magical food. Its own infection is actually the solution to a complete protein problem. Mind-boggling. Sure, fine. Okay, so that's amino acids. When the amino acids are linked together into chains, amino end to COOH end, it takes a little bit of energy input to make that happen. And for orderly chain forming, you also need an enzyme there to hold all the pieces in the right orientations. A hydrogen leaves the amino end and an OH leaves the carboxylic end. Those get together and form water, and we are left with 2 amino acids linked together in something called a peptide bond. And now we have a dipeptide. Keep linking on more amino acids, and now you have a polypeptide. Wait, you ask, didn't you say a string of amino acids is a protein? Where's this peptide nonsense coming from? Well, it's one of those not all rectangles are squares, but all squares are rectangles things. Most strings of peptides do grow up to be proteins, but before they get there, they have to be folded. Often a set of folded polypeptides then needs to be brought together and assembled to make a complete multi-unit protein. There are some single-unit proteins out there, but most are assembled from a set of folded polypeptides. Unlike fats and carbohydrates, which exist as more or less free, floppy, and relatively simple short molecule chains, proteins are organized. They have particular structures that fulfill particular jobs, and quite a few of them have 2 or 3 conformations based on local pH, local temperature, or microelectrocharge conditions. Of the 3 molecules, proteins are the ones that inspire the most dreams of micromanufacturing, 3D printing, and replicator technology. Those R groups have all sorts of active ends that do different things. There are positively charged ends, there are negatively charged ends, and there are neutral yet polar ends, and these are all sticky to water. Then there are the non-polar ends which repel water. The result is each of these chains folds up in a very particular way according to different R groups being attracted and repelled by either other R groups or the water-containing environment they exist in. Some protein chains are simpler and repetitive, like collagen, that makes up the framework of our bones, cartilage, ligaments, and blood vessels, plus all sorts of other connective tissue that holds our bodies together. Simpler protein chains are also responsible for fibers like hair, nails, horns, and the slime in hagfish, which is a scientific horror and wonder of its own special sort. More complex folded proteins mediate some of the most mundane, yet wondrous reactions that sustain life. A particularly well-studied example of this is hemoglobin, a complex of several proteins that puzzle piece together. They are able to pick up oxygen molecules as they pass along the capillaries that travel through our lungs. Then they hang onto them until they get to low oxygen areas where they change shape and release the oxygen to tissues that need it to carry on the activities of life. The shape is mediated by oxygen concentration, or pH, or a combination of the two. A slight change in the script for hemoglobin results in a protein that folds a bit differently, which means the red blood cells that carry it take on a different shape in one of their forms. And this is probably the best known example, since carrying one of these slightly off copies can actually offer an advantage when faced with the malaria-causing Plasmodium parasite carried around by mosquitoes. All this is to say, the intricately folded proteins take on very particular forms to perform very particular jobs. Holding us together and keeping us oxygenated are just a few of their jobs. Another big one is the role of enzymes. The chemical reactions that break apart food to release the energy we need to get stuff done depends on these twisty little guys. Humans are not constructed to withstand the other conditions under which food can be broken down to release energy. The other options are high heat, and humans depend too much on water as a liquid for that to work, or high pressure, and humans are a bit too squishy or too brittle for that to be an option, or long periods of time, and humans are too attached to our fairly quick rate of action. How do you think our ancient mammal relatives got away from the fermentation vats that were dinosaurs? So humans depend on enzymes, which are a class of proteins, to strategically destabilize bonds to allow the energy to get out quicker. And they allow for bonds to be made on purpose to build the stuff we need as well. The fact that we need enzyme proteins to make proteins is a problem similar to the it's much easier to make iron tools with iron tools, so how do you make the first iron tools to make an iron tool problem? Which means that at one point, all the assembly depended on haphazard stuff going on in primordial ooze, so it took a long time for things to get sorted out. Anyway, we are absolutely full of little wandering protein nuggets rushing about and doing important chemical jobs. But those free-floating proteins are relatively hard to find with our human eyes. The easy-to-find proteins are muscle, which of course is why we associate protein with meat, even though every living cell has some protein in it, and some specialized non-muscle cells are quite full of it, especially seeds. Under microscopes, muscles are made up of all sorts of fibers and bundles of fibers and strands of bundles, which is what gives muscle its thready aspect. The shape-changing aspect of proteins under different conditions is also what causes meat to change its characteristics so drastically when it's cooked. The process is technically called denaturing. When conditions are so extreme that these delicate little forms entirely lose their functional shapes forever, they've lost their nature. So we see denaturing when heat is applied, but it also happens in the presence of large pH changes. So acid baths. Think the citrus juice of ceviche, or exposure to bases, or what happens to cod in lutefisk, or eggs as pidin, or century eggs. Heavy salting also denatures proteins because it changes the charge environment in the water around the proteins and causes them to change shape and associations permanently. This denaturing habit, in fact, is what led to proteins as a specific substance getting identified in the late 18th century. The way egg whites, which are really just a protein gel, act in predictable but weird ways under heat, acid, and salts, that also turn up in other protein-rich goos. If you remember, egg whites were often leftovers in the making of fancy foods, so yes, they were definitely included in lots of early chemical experiments. If you're willing to mess about with them, egg whites are really rather fascinating substances. When there was no social media, people could get quite inquisitive about and involved in the study of odd phenomena. Denatured proteins have a common and particular texture, meat, egg whites, and other things, that is neither sticky, like carbohydrates, nor slippery, like fats. It's smooth, and it often forms strings, and while very flexible, it isn't very stretchy. It doesn't stretch much. And proteins, because of their nitrogen, sulfur, and some of the other complex R group components, can rot to produce some of the most varied of putrid smells. Look, early science was really grasping around for data to help them figure out patterns, so, they recorded a lot of weird stuff. But let's address the questions at the top. 1. Why are some muscles stringier than others? Different amounts of collagen, that connective tissue wound in with the other muscle fibers. The more active and the more different directions a muscle has connections, the more collagen is formed in and through it. So muscles that do a little work, less collagen is incorporated. That also means younger muscles have less collagen, which is why most of the meat animals in America are incredibly young at slaughter. So they'll be tender, which is also why we have bred most of our meat animals to gain size quickly. There are some downsides of this, but the one that matters here is that they lack flavor. Rapid growth requires that there's no complexity to the tissue, so there's not going to be any interesting flavors. In quadrupeds, this means that the neck, chest, and leg, especially lower leg muscles, are going to be the stringiest. But back and belly muscles get the least work, so are the tenderest. In avians that fly, all muscles are going to be rather stringy. Being a wild bird is hard work, and those animals are so stripped down for flight, there's no space for muscles that aren't doing anything, which is why chicken and turkeys are so crazy. As basically flightless, they just have these breast muscles on board for display, so that's why it's so tender and light, and why it ends up being called white meat, because it lacks the capillaries to bring in enough blood for them to be properly strong. Number 2, jellyfish. They appear in food as they do because they are basically elegant collagen structures with a few strips of muscle and some organs. As with humans, they can have parts where the collagen is less flexible due to the amount of incorporated water or minerals. They can also contain particularly rigid bits of chitin. The most striking examples of this are the velella, or that sort of jellyfish that have their own sails. Check the show notes for a link to a picture of these iridescent blue space creature-looking things. Okay, technically, chitin is an amino sugar complex, so technically a carbohydrate, and is a bit closer to the polysaccharide cell walls in plants, but it's still an example of a long-chain amino compound. Biology is full of confusing exceptions and so many categories, no wonder it has so many specialties. Anyway, because the boingy parts of jellyfish are just very hydrated collagen, they can be easily dehydrated and then rehydrated to make boingy, firm, chewy noodles with a bit of satisfying snap as your teeth go through them. Number 3, we associate umami and meaty because one of the plentiful amino acids found in meat proteins is glutamic acid. And when we dehydrate muscle meat through cooking or drying or aging techniques, the glutamate is released. For reasons, we have a taste apparatus that recognizes glutamate specifically, and for other reasons, most people find the chemical sensations we register as taste pleasant. But that amino acid hangs out in other places and sometimes just as an amino acid, not all tied up in a protein. Tomatoes, mushrooms, and walnuts are all examples of such places, which is why they often get the label umami-rich. Number 4, more mushrooms. Plants and mushrooms, can they be sources of protein? Sort of. Sure, they have some proteins all living things have to in order to hang together, but what mushrooms and plants really do is they bring along a whole bunch of amino acids, which, let's face it, are way more useful for us as the raw materials for protein. See, we can't actually use proteins as they arrive in our bodies from other organisms. We have to bust them apart into smaller units and then reassemble them as they're needed. Which is why vegan and vegetarian diets can be perfectly helpful when people are sensible about them and not using them as elaborate ways to restrict food intake or disguise disordered eating. So as long as you don't just eat only PB&Js or only corn chips and guacamole, you'll be fine. While it is true that the busting apart of proteins into individual amino acids is a source of energy, I mean, We have to add energy to make peptide bonds, so you get energy out when you break them. That's where the energy comes from eating protein. Refer back to bonus episode 69.5 on ice and fire science for more on bond energy. But the energy coming out of busting peptide bonds is smaller when compared to the energy found in the breaking of bonds in fats and carbohydrates. That, incidentally, is the science that's underneath the how many calories in a gram of protein or in a gram of fat or in a gram of carbohydrate label on those exhaustively complete nutrition labels. While there's some argument about the absolute amount of calories, the relative amounts are sound enough. Which brings us to point 5. Can you almost live on bread alone? If you go back to the rice and wheat and corn pairings, each major grain culture has already figured out the best buddies for their favorite grain to get us the complete set of 9 missing amino acids we need. So sure, you can't just live on bread, but bread or rice or corn, but also yam, cassava, teff, potato, and all sorts of other basic starches with their proper toppings will do you all sorts of good. So just look at what any given culture pairs with its favorite starches, and yeah, you can bet that all 9 of those missing amino acids are there. And that approach makes for a way to explore the world of taste. In fact, the more different tastes you chase, the more likely you are to be getting all the stuff you need in the amino acid or protein department. All those strangely folded little protein molecules and their amino acid puzzle pieces are some of the most interesting flavors out there. Thanks for listening. Any email, questions, or quibbles will reach me at thehistoryofamericanfood@gmail.com. Shout out, as always, to the Dinosaur Neil episode of The Tick. No unnecessary innovation, my foot. And so please support the funding of public television shows that link kids to science, curiosity, and critical thinking. Music for this podcast is Fingerlympics by Dr. Turtle. The link to the show notes and other ways to send me questions are also in the show's Twitter, Insta, and post.news bios at THOA Food and in the show description.